Affinity chromatography takes advantage of the fact that many biological molecules naturally recognize and bind to specific partners. The column is designed with a ligand, such as a metal ion, a sugar, an antibody, or a DNA fragment, attached to the resin, and only molecules that have the matching binding site will stick. Everything else flows through. Once the contaminants are washed away, the target molecule is released by changing the buffer conditions, usually by adding something that competes for binding or by shifting the pH or salt level. Because the separation is based on a highly specific interaction, affinity chromatography can isolate one protein out of a huge mixture with very high purity, often in a single step. This is why it's so widely used for purifying recombinant proteins, tagged proteins, and important biological complexes. Some examples include antibodies and antigens, lectin-based affinity, which binds particular glycoproteins, nucleic acids with complementary bases, hormones with receptors, and immobilized metal ion affinity chromatography, which uses a his-tagged protein that binds nickel 2+ on a chelator resin. A his tag is a short amino acid composed of histidine residues that is added onto a protein at its C or N terminus. Such uses of affinity chromatography have revolutionized downstream processing in biotech. What used to take multiple steps, such as precipitation, ion exchange, then ultracentrifugation, can be done with one affinity capture step. Some caveats, though, are that it is usually more costly, often requires development of the ligand, and some elution conditions might stress the molecule. In the previous section, I mentioned diseases being treated by monoclonal antibodies, purified with ion exchange chromatography. I actually found that many of these same treatments involved an affinity chromatography step beforehand. In the treatments for both breast cancer and inflammatory diseases such as rheumatoid arthritis, there's a protein A capture step, wherein protein A is immobilized on resin and binds to immunoglobulin G therapeutic antibodies. Affinity chromatography also, like IEC, plays a role in the treatment of diabetes, specifically recombinant insulin and analogs of insulin. Before modern polishing steps became standard, immunoaffinity and peptide affinity were used to purify insulin. The ligand used was anti-insulin antibodies and insulin-binding peptides that were fixed to the column resin. Thanks to this method, misfolded or truncated forms of insulin could be removed, which was necessary to avoid immune reactions and hypoglycemia. Additionally, affinity chromatography has been used in the treatment of hemophilia A and B, making what was once a fatal disease a treatable but chronic condition. Chromatography has allowed for purification of recombinant and plasma-derived clotting factors, with factor VIII and factor IX proteins binding to their respective anti-factor immobilized ligands. As I was researching affinity chromatography further, I was excited to discover that it is responsible in part for the CRISPR gene editing tool, which I was completely fangirling over. CRISPR itself is a system bacteria evolved for immune defense that we as humans have co-opted for our own gene editing purposes. In order to do this, we needed milligram amounts of highly purified Cas proteins. A method of achieving this used enriched Cas9 with a C-terminal his tag from E. coli lysate with nickel 2+ NTA metal affinity chromatography. Without affinity chromatography, we would be unable to purify big multi-domain nucleases such as Cas9 and Cas12 at the scale desired. The final use of affinity chromatography I will discuss involves oncogenes and tumor suppressors. Immunoaffinity chromatography was done to purify EGFR, the epidermal growth factor receptor, from A431 carcinoma cells. The ligand on the column was anti-EGFR monoclonal antibody, and the binding molecule was EGFR, which is an oncogenic receptor tyrosine kinase. With this chromatography, it became possible to quantify the receptor number and autophosphorylation, as well as design EGFR-targeted inhibitors and antibodies to be used in cancer therapy. As promised, I will also discuss tumor suppressors, namely the p53 protein. A rapid immunoaffinity column purification was used to isolate murine p53 chaperone complexes, which allowed us to analyze HSC70 DNA-K interactions, bettering our understanding of how p53 tumor suppression works. After this, highly purified p53 was used to show that p53 has intrinsic 3' to 5' exonuclease activity, also using affinity chromatography with anti-p53 antibodies at the ligand. With all this knowledge acquired through chromatography, much work has been done to understand how DNA damage responses work, how p53 mutations can cause cancer, and how to design inhibitors and antibodies against these pathways. I am unsurprised by how many of our modern biotechnologies owe their existence to affinity chromatography, as its specificity makes it such a powerful and targeted tool. Having learned all of this, I'm ready to discuss the final method of hydrophobic interaction chromatography.